Hello students, welcome to EPG Pathshala. I am Dr. Gayatri Ramakrishna from Institute of Liver and Biliary Sciences. Here we are going to discuss the module Basics of Cancer Biology from the paper Molecular Therapeutics. Basically, this module will cover the 5 objectives: the characteristics of a cancer cell, the cell cycle control, the causes of cancer, the various genomic instabilities, and finally, the various hallmarks of a cancer phenotype. This module will try to cover some important aspects on cancer cell biology. Basically, the module will discuss in detail the various characteristics which make a normal cell deviate towards a cancerous state. This module will first describe the importance of a normal cell cycle. Further, we will see how deregulation will lead to abnormal growth characteristics of a cancer cell. In addition, we will also briefly cover the various causes of cancer, and these etiological factors will include a brief description on the chemical agents, the physical agents, and the various viral factors which contributes to the process of carcinogenesis. In addition, we will discuss how cancer cells become immortalized. This in turn will cover the basic aspects of the telomere biology, the activation of the telomerase, which is important for maintaining the telomere length and thereby preventing the process of aging. Additionally, we will also discuss the different types of the epigenetic and the genetic alterations, and these in turn include the various mutations in the cancer drivers, namely the oncogenes and the tumor suppressor. We will also discuss briefly the microsatellite instability, which is yet another important hallmark feature of many cancer types like the colorectal cancer. Finally, the module will introduce some landmark papers which discusses the hallmarks of cancer. We will discuss the 6 different rules which makes the cancer phenotype. Besides the 6 basic characteristics, we will also discuss the emerging hallmarks such as the reprogramming of the cellular metabolism and also the immunomodulations, which are the underlying features of cancer. Basically, cancer is nothing but deregulated cell cycle. In fact, cancer is basically a genetically dictated loss of cell cycle. The progression from a normal to an abnormal phenotype of cancer includes multiple events. Hence, cancer is not a single event process, rather it's a multi-step event with multiple hits in the oncogenes and the various tumor suppressors. Let us understand how cancer is a deregulated cell cycle event. Let us imagine a group of cells in which one cell becomes abnormal and is denoted by a red ball and start to proliferate uncontrollably. In course of time, the abnormal red cell will take over the population and spread to other parts of the body. Thus, cancer is basically a genetically dictated loss of cell cycle. In fact, the progression of the abnormal cell to become fully malignant involves multiple step events. To understand what controls the cell division, one needs to understand the various phases of cell cycle. The cell cycle consists of 4 phases. Following the mitosis, the cell enters the first growth phase or the G1. The cell diploidy is 2n. Most of the adult cells in the body are in the G1 phase. At the G1 phase, a decision can be made to exit the cell cycle and the cell remains dormant or quiescent. This stage is referred to as the G0 phase. When cells have to proliferate, they enter the S phase. This is the phase of DNA synthesis and the ploidy of the cell is more than 2n. Once the DNA synthesis is over, the cells enter yet another growth phase, which is a preparatory phase for the cells to divide and is called the G2 phase. In the G2 phase, the ploidy is 4n. In the mitosis phase, the cells initiate to divide, the chromatin condenses to chromosome, and there is a separation of the daughter chromosomes, and finally cytokinesis gives rise to 2 daughter cells. Usually, cell cycle is controlled and not all cells at a given time are dividing. As an example of controlled division, when there is a cut in the skin, the gaping wound is filled by the dividing cells. During burns, when there is a loss of cells, the skin cell can regenerate. Similarly, liver injury, almost half of the liver has the capacity to regenerate. These are all controlled examples of cell cycle regulation. However, in cancer, there is an uncontrolled proliferation and the cells keep dividing and the cell cycle is deregulated. One important aspect that governs the division is the contact inhibition. Cells cease to divide when they make cellular contacts, as is often seen in an in vitro cell culture system. However, in cancerous condition, cells do not obey the rule of contact inhibition and keep on proliferating. So let us examine some characteristics features of the cancer cell. As already explained, the first basic feature is that the cells have lost the ability to stop dividing, and this makes the cells immortal. But still, not all immortal cells are cancerous, as immortal cells, when following the rules of contact inhibition, can stop the growth. However, immortal cells which have lost the ability to undergo growth arrest will keep on dividing and are hence called cancerous. Yet another important characteristic feature of cancer cell is metastasis. That is, the cells can migrate out from the tissue of origin and spread across the body through circulation and can home in other organs. For instance, liver cancer can metastasize to bone or lung. The most important characteristics of most of the cancer is its heritable property, because cancer cells arise by mutation in driver genes such as the oncogenes and tumor suppressors, which usually show loss of their function. And the examples are like the p53 and the retinoblastoma gene. This slide shows the basic characteristics of the cancer cell, which includes deregulation of the cell cycle, activation of the telomerase leading to immortalization, the genetic and the epigenetic alterations leading to increased cellular proliferation, and most importantly, the loss of contact inhibition leading to metastasis and spread of the cancer. This slide elaborates yet again the process of cancer progression and the fail-safe mechanism. Whenever there is a DNA damage, the cell cycle checkpoints are activated. And if the DNA is not repaired, it will lead to mutations or deletions, thereby changing the proto-oncogene into an oncogene. If the DNA is not repaired as a safe pathway, then the cells take an alternate pathway of cell death. However, cancer cells avoid this and show reduced apoptosis. And this leads to continued cell proliferation and abnormal growth. Yet another fail-safe mechanism is to prevent the telomerase activation. And when telomere shortens, it leads to aging or the process of cellular senescence. However, during cancer, the cancerous cells overcome the process of senescence and hence regain a more youthful-like phenotype because of increased telomerase activity. Finally, contact inhibition, as was explained in the earlier slide, is the key to control growth. But loss of contact inhibition not only leads to increased tumor mass, but also the cells from the tumor are shed into the circulation and they can spread out to other parts of the body. This slide gives a brief overview of telomere biology and how it is linked to the process of cancer. The telomeres are the end of the chromosomes, and with each round of division, the telomeres shorten. A very critically shortened telomere is perceived as DNA damage and leads to crisis. During cellular crisis, the cells undergo either a process of senescence or they eventually die. However, at crisis, few cells initiate or activate the telomerase, thereby maintaining the telomere length. And these are the cells which then become immortal. And in the later steps, these immortal cells will have more genetic or genomic instabilities, thereby converting them into a malignant cell. Let us now understand how genetic instabilities arise. Let us understand something about the genomic instability. We will cover two aspects: what causes the genomic instability and what all different types of genomic instabilities exist. Genomic instabilities is basically due to certain carcinogens. These carcinogens can be either a chemical carcinogen, or it can be a physical carcinogen, or it can also be viruses. The most important example is the cigarette smoke, which is rich in polyaromatic hydrocarbons. And similarly, heavy metals like arsenic can also lead to genomic instability. Besides chemicals in environment, other physical agents such as the radiation, especially the UV radiations or the X-rays, can also be damaging the DNA. Yet another causative factor are the viruses, which includes both the DNA and the RNA virus. In fact, in India, Indian women, cervical cancer rate is very high, and the cause is the human papilloma virus. Similarly, the liver cancer or hepatocellular carcinoma is caused by the hepatitis B virus. Once the cell becomes immortal, as we had earlier seen by an activation of the telomerase, there are events that lead to accumulation of genomic instabilities. In fact, the cause of the cancer can be divided into genetic alterations, which include the DNA mutations, the microsatellite instabilities, and the chromosomal instabilities. Most DNA mutations are spontaneously generated because of an inefficient DNA repair. However, some mutations are familial, that is, they are passed from one generation to another. For example, the mutant BRCA gene is passed from the mother to the daughter. Most of the mutations affect the protein functionalities. In addition to genetic alterations, there is yet another alteration which is called as the epigenetic alteration, which is mostly caused by the environmental factors, and majority it includes the hyper or the hypomethylation of the CpG islands in the promoter regions of the gene, thereby causing changes in the gene expression pattern. Why should these genomic alterations happen? As we had already seen in the previous slide, the DNA damage is most commonly caused by chemicals like the cigarette smoke, the heavy metals, the radiations, etc. These can damage the DNA by either causing something like a thymidine dimer or by the formation of the adducts. And whenever such DNA lesions occur, they have to be removed. This removal takes place by the DNA repair system. However, in the case of cancer, the DNA repair itself is defective, and this defect gets fixed in the daughter progeny, thereby fixing the mutation in the resulting progeny. This slide shows the DNA damaging agents, the common lesions, and the affected DNA repair pathways which are mostly affected in cancer. Also shown here in red are the genes and the pathways which are epigenetically regulated to have reduced or increased expression in various cancer. the various DNA repair pathways that exist, and that leads to an error-prone system. However, any defect in this error-prone system will lead to inclusion of common lesions in the DNA, leading to genomic instability. As an example, one can see the different genes which are involved in the DNA repair pathway. For instance, the gene MGMT is O6-methylguanine DNA methyltransferase, which repairs the naturally occurring mutagenic DNA lesion O6-methylguanine back into guanine. Similarly, the gene MLH1 is associated with hereditary non-polyposis colorectal cancer or HNPCC. And this gene product prevents the mismatch and errors during DNA replication, and this gene is often mutated in colorectal cancers. The various cancer drivers. For instance, these include the oncogenes and the tumor suppressors, which are frequently mutated in various cancers. Oncogene is like an accelerator and tumor suppressor is like a brake in a vehicle. Proto-oncogenes are required for normal proliferation. However, whenever there is a mutation in the proto-oncogene, it leads to hyperfunction of the gene. For instance, KRAS is frequently mutated in lung and pancreatic cancer. And in fact, KRAS is a signal transducer which helps in cellular proliferation. In addition to mutations, gene can also be amplified. That is, for instance, the gene MYC, and excessive MYC can deregulate cell cycle. On the other hand, tumor suppressors are the brakes of the cancer. Mutations in the tumor suppressor gene often leads to loss of function. The best known examples are that of p53 and the retinoblastoma. Another important aspect of the tumor suppressor is described by the Knudsen-2 hypothesis, which involves loss of heterozygosity when one gene is mutated, and when both the genes get mutated or they are lost, it leads to loss of function. As shown in this diagram, one white normal allele and one red mutant bar indicates loss of heterozygosity. When the second hit happens, both becomes the mutated or the red bars, resulting in tumorigenesis. In yet another case of familial, one mutated red bar is inherited from the parent, and another occurs during their lifetime, again leading to total loss of function. This is best described by the example of retinoblastoma, which is a childhood eye tumor. What causes the oncogene activation? The various reasons for the oncogene activation include point mutation, amplification, translocation, and epigenetic changes such as the hypomethylation of the genes. Besides the genetic alterations, such as mutations and deletions, yet another hallmark feature of the cancer is microsatellite instability, or MSI. So in next few slides, let's learn what MSI is and what is its utility. Microsatellites are small tandem nucleotide repeats which can be either a mononucleotide or a dinucleotide or a trinucleotide, which can be present in either an intron or an exon. Mostly, microsatellites are polymorphic in nature. Hence, they have various utilities, such as in population genetics, in DNA fingerprinting, and especially in case of cancer, it can be also useful for diagnostic and prognosis. Microsatellite instability occurring in cancer can be either gain or loss of the tandem nucleotide. As is shown in this example, let's imagine a normal state where a dinucleotide CA is repeated 10 times. However, in a disease state like cancer, this CA repeat can be either more than the normal, that is more than 10 times, or it can be less than 10 times. Thus, there can be either an expansion or a loss of the dinucleotide, thereby leading to pathogenic events. This loss or gain of the tandem nucleotides results in allelic disbalance, which is often associated during cancer initiation. Let us understand this by an example. This is just an example of how the expansion of the repeat D17S250 loci is seen in a patient in a cancer versus a normal. As you can see, the blue peaks actually represents expansion of a repeat in the cancerous region, whereas it is normal and limited in the normal sample. This slide describes how MSI can lead to cancer progression. For instance, a normal microsatellite loci can expand or contract due to either a DNA polymerase slippage or defect in the mismatch repair genes, leading to their expansion or contraction. If this occurs near the site of an oncogene, such as a RAS, or a tumor suppressor such as a P53, then it can lead to genomic instability, which can make the cells escape from cytotoxicity to drugs. Hence, MSI can be used in cancer diagnostics and prognosis. This slide shows how microsatellite analysis is useful in cancer prognosis. A panel of microsatellite instability markers is used in cancer diagnosis, especially in the colorectal cancers, where one uses a combination of at least 5 MSI markers, which include mononucleotide repeats like BAT25 and BAT26, and 3 dinucleotide repeats called the D2S, D5S, and the D17S. Several studies demonstrate that patients with high microsatellite instability status respond better to surgery alone rather than chemotherapy and surgery together. Thus, microsatellite instability analysis helps in delineating patients who can undergo directly surgery without experiencing chemotherapy. Hence, a prognostic value to MSI. Yet another genomic alteration is the epigenetic changes in the cancer. For this, we need to understand what an epigenome is. The epigenome is described as same nucleotide sequence, but modification of the chromatins in form of CpG-island methylation, histone modifications such as acetylation, and yet another aspect is the nucleosome architecture. All these 3 different forms of modification on the chromatid determines whether a gene is actively transcribed or silenced. Thus, epigenome affects the gene expression. Recently, whole exon sequencing of human cancer showed striking differences in the epigenome, as there are high proportions of methylation changes and different histone acetylation patterns, thereby causing gene expression variations in cancer vis-a-vis the normal tissue. For instance, as shown in the lower figure, a hypomethylation of the oncogene causes gene activation, and hypermethylation of the tumor suppressor causes loss of their suppressive function. This slide shows the cancer epigenome landscape. The top figure illustrates the various epigenetic changes such as the hypermethylated, and also the epigenetic targets in cancer progression. The lower figure is a hypothetical scheme that explains how epigenetic mediators might reprogram the epigenome to tip over the normal somatic cell or a differentiated progenitor cell into a cancer stem cell state, displaying phenotypic heterogeneity. Having understood the genetic and epigenetic alterations, it is clear that many genes are involved in the genesis of cancer. In fact, cancer is a complex phenotype and involves an evolutionary process which includes the process of initiation, promotion, and progression. Further, alterations both in genome and epigenome during these 3 steps lead to a fully metastatic pathological entity as a cancer. Let us see this with 2 examples of colorectal and liver cancer. This is an example of genetic alteration and progression of colorectal cancer in a sequential multiple hit process. The major signaling pathway that drive tumorigenesis in a colorectal cancer are shown at transition between each tumor stage. And as is noted, the progression of the colon cancer occurs in many years, and the driver gene is the adenomatous polyposis coli gene or the APC gene, which causes the cancer initiation. This is followed by activation of other genes such as the RAS, genes like the PI3 kinase, TGF-beta, which completes the evolution of the colorectal cancer. This slide tells in a nutshell the various features underlying the liver hepatocellular cancer progression, which again involves genetic, epigenetic heads. In addition, as you can see, there are certain underlying hallmark features such as telomere stabilization, inflammation, angiogenesis, etc. during the cancer progression. This has been very nicely described by Weinberg and Hahn, and they have come up with a landmark paper on hallmarks of cancer as shown in the next two slides. Hahn and Weinberg, in the classic paper called The Hallmarks of Cancer, describe rules in the transformation of a normal cell to cancer cell. And this includes 6 major basic features. Number 1, self-sufficiency in growth signals. Number 2, insensitivity to growth inhibitory signals. Number 3, envision of programmed cell death. 4, limitless replicative potential. Number 5, sustained angiogenesis. And number 6, tissue invasion and metastasis. The next generation hallmark, besides including the basic 6 hallmarks, include more emerging hallmarks. And the 2 hallmarks which are important in this regard are 1, reprogramming the cellular metabolism, and the other is the property by which the cancer cells evade the immunological destruction. Additionally, 2 consequential characteristics of neoplasia facilitate acquisition of both core and emerging hallmarks. And this includes the genomic instability plus the inflammation by the innate immune cells. Thus, these emerging hallmarks are now appreciated as yet another underlying feature of the genesis of cancer. Dear students, we will now summarize what we have learned today in this module of cancer cell biology. We have learned in this module that cancer is a multi-step process which basically involves 3 basic steps of initiation, promotion, and progression. There are various etiological risk factors for cancer, such as chemicals, for instance, from smoking cigarette, radiations like the UV and the X-rays, viruses such as the human papillomavirus, the hepatitis B virus, the HIV virus, etc., that can cause deregulation of the cell cycle and genomic alteration. Cancer cells have deregulated cell cycle. That is, they have lost the property of growth arrest. Not only that, they also escape cell death. From a normal cell to become into a cancerous, the first step is immortality. And this is achieved by activation of an enzyme called the telomerase. The telomerase enzyme helps in maintaining the telomere, which are at the end of the chromosome. And telomerase prevents the telomere attrition, and thereby it prevents growth arrest and also the phenomenon of aging. Once the cell becomes immortalized by activation of the telomerase, this is then followed by a series of genetic and epigenetic alterations. In fact, there are various accumulations of these changes, which then make the cell, a normal cell, to acquire a neoplastic phenotype. The various genetic alterations include mutations, deletions, amplification of the genes, microsatellite instability, and chromosomal instability. In addition, there are also epigenetic alterations. This includes basically a chromatin remodeling by methylation, histone acetylation, and nucleosome arrangement, which in turn leads to differences in the gene expression profile. Most of the genetic alteration occurs in the driver genes. These driver genes include basically the oncogenes such as the KRAS, the RAF, and the MYC. Besides this, there is another important class of genes called the tumor suppressors. Tumor suppressors act as the brakes, while the oncogenes act as an accelerator. Thus, when oncogenes get activated and when there is a loss of function in the tumor suppressor, this leads to a process of multi-step event leading to oncogenesis. Genes like KRAS, P53, RAS, and MYC are the most common cancer drivers. Besides these genetic alterations, there are 6 hallmarks or underlying features in the process of carcinogenesis. These 6 features include self-sufficiency in growth signals, insensitivity to growth inhibitory signals, avoiding cell death, limitless replication, angiogenesis, and tissue evasion leading to metastasis. Besides these 6 underlying features, recently 2 more emerging hallmarks have been added, and this includes reprogramming of the cellular metabolism and evasion of the immunological destruction. The genomic instability and inflammation are 2 important drivers of cancer progression. So dear students, we will now end the module once again recapitulating the summary. So to summarize, cancer cells show deregulated cell cycle. There are various chemicals and physical entities, including viruses, which are important etiological risk factors for cancer. Cancer cells are all immortal, and this immortalization is through activation of the telomerase and maintaining the telomere length at the end of the chromosomes. Finally, cancer is heritable, which includes genetic and epigenetic landscape changes from a normal to a cancerous phenotype. And last but most important, cancer is a multi-step process with certain hallmark features like angiogenesis, immunological evasion, etc. Thank you.